Oh, all right. Hey guys, going on? My Caveman Q here again. Paleo Palm Island, mycaveman.com. Got another edition of One Nights with Mike. Live stream Q&A. Ask me anything, health related. You know the deal. And you know what? Not really crazy about the lighting right here. Creating a weird look right here. All right, it's mildly better. All right, so you know the deal. Questions, comments, put them on down over there or down below, depending on what kind of viewer you're viewing this on. And while you do that, we're going to be taking some other random questions that, well, not random, but other things I've been asked about recently, and we're going to look at through some of the research that has been done on those topics. So, some of the things we talk about today are the types of senses that we have when it comes to taste. So, we have our most common ones. We have salty, sweet, sour, bitter, and umami. But is fat also a potential taste? We'll talk about that a little bit. And then from there, we're going to talk about how that plays into the neuroregulation of appetite, or basically how different foods affect the way we regulate how much food we're going to eat, how much, whether we're going to eat more or less. And then from there, we're also going to look at a relatively recent study that's been making the rounds with respect to the use of an autoimmune protocol in dealing with autoimmune disease. Specifically, in the case of this study, it was done on IBD, or irritable bowel disease. So let's get started. So first one we're going to talk about, like I said, is going to be with respect to different taste buds and different taste receptors, rather. So a taste of fat, a sixth taste modality, is what Besnard, Pacilli de Grace, and Kahn asked last year in 2016. And looking through that, so an attraction for palatable foods rich in lipids is shared by rodents and humans. And over the last decade, the mechanisms responsible for this specific eating behavior has been actively studied. And compelling evidence implicates a taste component in the orosensory detection of dietary lipids, i.e., long chain fatty acids, in addition to the textural, olfactory, and post ingestive cues. The changes that affect both food intake and digestive function. The interaction between long chain fatty acids and specific receptors in the taste bud cells elicit physiological changes that affect both food intake and digestive functions. After a short overview of the gustatory pathway, this review brings together the key findings consistent with the existence of a paradigm, i.e., chemoreception of long-chain fatty acids in the taste bud cells, cell signaling cascade transfer lipid signals throughout the gustatory nervous system, and physiological consequences. They will be critically analyzed. The limitations to this concept will also be discussed in the light of our current knowledge of the sense of taste. And finally, we will analyze the recent literature on obesity-related dysfunctions in orosensory detection of lipids, or fatty taste, in relation to the overconsumption of fat-rich foods and associated health risks. So, can fats cause you to overeat? Hey, Jody, what's up? Um, Now, we have a couple of other papers that we're going to look through on this. This one's a particularly long one. It's about 20 pages long. Unfortunately, it is behind a paywall, so we're not going to go through the whole thing today, and it will be a little bit tougher for you to get access to it. However, if you are so inclined as to read the whole thing, let me know. I will try and get it over to you. But here we go. So obesity is one of the major public health challenges of the 21st century. And on account of its rapid worldwide spread and the deleterious effects of associated diseases, i.e., non-insulin dependent diabetes, cardiovascular spread, cardiovascular disorders, hypertension, cancer, and neurodegenerative disorders, profound changes in our lifestyle, especially in our eating habits, are thought to play a significant role in this phenomenon. The availability of highly palatable and energy-dense foods rich in fat and sugar has been associated with with the risk of developing obesity. The hedonic reward value of these foods constitutes a driving force promoting their preferential consumption. The fact that energy-dense foods are attractive and thus preferentially consumed might constitute an advantage inherited from evolution to survive a food scarcity. Together, these observations suggest the existence of an early and selective detection of these caloric nutrients. This sensing occurs as food enters the oral cavity and depends on the effectiveness of three complementary sensory systems: the synesthesia, i.e., mechanoreception, thermal sensation, and nociception; the retronasal olfaction; and gustation, The orosensory perception of dietary lipids, in contrast to sugar, was long thought to depend solely on their textural and olfactory properties. The recent discovery of lipid receptors in rodents and human taste buds suggests that lipids might also be detected by the gustatory pathway. Is there a sixth taste modality devoted to the detection of dietary fat? Neuroimaging studies suggest that the reward circuit could be disrupted in obesity. Does the obesity also lead to dysfunction of the orosensory detection of dietary lipids? What could be the impact on food health choice? Current knowledge on these issues will be discussed in this review. So, short version on what the hell that all just said. Basically, like I said before, there are 5 primary tastes that we think of when it comes to our taste buds. There's salty, which is our body's ability to detect sodium and or other electrolytes like that. There is our ability to taste sour, which is tasting for free hydrogen ions or basically the acidity. There is bitterness, which is mostly tasting for alkaloids. There is uh, sweet, which is tasting for glucose and glucose analogs, other sugary analogs. And from there, we have umami, which is the weird one that you don't learn about until you're a little bit older, which is basically our body's ability to detect for free glutamate, and that is most commonly associated with MSG or monosodium glutamate, but it refers to that savory taste. So we have detectors for protein, for carbohydrate, for um, for electrolytes, and for alkaloids. Why wouldn't we have one for fat? Well, up until very recently, try to say relatively and then very at the same time. But up until very recently, we hadn't necessarily discovered the suggestion of lipid receptors. Well, recently we have, particularly the CD36, but we'll come back to that later in another paper. But yes, or maybe later in this one too, I believe so. But we're going to breeze this one quicker. Anyway, the fact of the matter is, they have been discovered now. So it quite possibly is that fatty is another taste. Up until then, we had thought that it was basically based on both the taste, or rather, the texture, so the touch sensory, both on the tongue and the mouth of fats, basically that traditional oily, fatty feel, the mouth feel, as well as olfactory or smell related properties. So we had previously assumed that, or scientists had previously assumed that smell and touch were the factors that allowed us to detect fat, but now we're seeing that it actually might be a taste component. From there, we're talking about neuroimaging studies, talking about the reward pathway, how it could be disrupted in obesity. Basically, that in people that are obese, that ability to taste fat may be disrupted. There may be a chink in the transmission of the information to the brain from the tongue. From there, we have talked about how this could actually change whether we are able to regulate our food intake. There's always the calories in, calories out model where just eat less food. Well, maybe both our hormones and our ability to detect them, so our neurotransmitter, our neurotransmission pathways are disrupted. So, short overview of the taste system. Like I was saying, basically, the sense of taste provides critical information about the quality and nature of food, leading to specific eating responses, consumption, or avoidance. 5 basic taste modalities recognized are sour, salty, sweet, bitter, and umami. The related taste stance are detected by taste buds mainly localized on the tongue epithelium and rarely in the soft palate, pharynx, and upper esophagus. In the lingual epithelium, taste buds are present in 3 specialized gustatory papillae, i.e. the fungiforms, the foliates, and the circumambulates. Yeah, we're getting a little deeper than we have to. But anyway, they're displaying different spatial distributions. Most of the taste buds are found in the circumambulate papillae. They are characterized by the dome-shaped structure with the circular depression connected to the von Ebner's glands, which produce and release salivary enzymes. So here we go. We have our circumambulates. That's generally that back section of the tongue is where most of our taste is coming from. Those dietary compounds, they come in, they go down all the way to our taste buds. They get broken down through those various promoters there, and there we go. Moving forward, taste bud as the primary receptor organ, studies on taste buds reveal an unexpected complexity. First, each taste bud is a heterogeneous assemblage of 3 different mature elongated cell types, types 1 through 3, and 1 basal cell type, 4. Type 1 cells are glial-like supporting cells, sharing many features with astrocytes. They are thought to participate in sensing sodium. Type 2 are taste receptors cells expressing in mutually exclusive manner the receptors for bittersweet and umami. They communicate with afferent gustatory nerve fibers through non-conventional synapses. Type 3, and then if we pull this back for a second, you get to see a little bit of this in the figure. No, sorry, this is figure 2. This is the distance. These are the tastants. So type 1, type 2. So type 1 on the outside, type 2 are the middle ones, type 3, these nice little yellow ones, and type 4 are these little guys on the bottom. So they're the fibers, and then they're the circle at the bottom. Okay, so type 3 are presynaptic cells required for sour taste perception. They form synapses with the primary sensory afferent terminals, and then type 6 and or 9 of the cranial nerves. And then finally, we have type 4 cells, which are precursor cells responsible for taste bud cell renewal. So they help to rebuild those. Then we have here the peripheral coding of taste is still debated with two proposed theories. In the label-lined model, the distinct taste qualities are carried by one type of TBC connected to dedicated afferent nerve fibers, while in the combinatorial model, also called the across fiber model, a taste modality is encoded by several broadly tuned neurons. So moving forward, that's the basic idea on it. We see here, basically, we have the different types of the hormone functions and the taste buds. Sorry, rather different hormone location and functions in the taste buds. Are they in type 1? Are they in type 2? Are they in type 3 and type 4? What do they do? Okay. Are they present? What do they test? They taste an upregulation of sweet and bitter, downregulation of sweet, upregulation of sweet. Okay, so that's basically the different types we have going on. From there, from taste perception to food selection. As aforementioned, the connection of lingual gustatory gustatory papillae to the central nervous system occurs via two cranial nerves, the fungiform and the circumferent papillae. They are innervated by the CT, the cranial nerve number 7, and the GL, cranial nerve 9, the afferents, respectively, while the foliate papillae are innervated by both. The afferent fibers convey taste information to the rostral nucleus of the solitary tract, the RNST, in the brain stem, which is the obligatory first synapse in the ascending gustatory pathway. All right, let's get past what we really want to focus on for right now. So basically, food comes in. We have our energy nutrient. Goes to our taste bud cells. That goes and releases hormones. Those hormones then go and send off information to neurotransmitters, which then in turn send off that information to the NST, which then can either initiate or stop digestive function. From there, that sends it to the central nervous system. And we start looking about the palatability. Do we like it? Okay, yes, we do. We learn that we want, so we learn that we like it. From there, we say, yeah, we want more of this. So this is in the decision selection method. From there, we get to how much energy does it provide? Provides a lot of energy. Our metabolic brain, so the part that says, hey, we need energy and lots of it often says, yeah, good thing. So we have our emotional brain. Does it taste good? And our metabolic brain. Will it help us survive? If we pair both of those together, it says, yes, eat more of that. Okay, so the VTA, the NAC, the amygdala, the hippocampus, and the OFC all contribute to whether or not we like that food. From there, we have our hypothalamus helps contribute that to whether or not it's going to help us live. Okay, then we have down here extra nutrient sensors. So in our small intestines, once that energy actually makes it all the way down to our small intestine, we send out enteroendocrine cells. So our stomach, or rather our small intestine, sends out information, which then in turn sends out hormones and neurotransmitters. Those neurotransmitters can in turn send information back to that pathway to help determine whether or not it's a palatable food, whether we like it or not. And it sends off hormones over here, which in turn help your body to know whether or not there was glucose or lipids taken in. Those will help to form metabolic signals from the digestive tract and other organs, such as adipose tissue and endocrine pancreas, basically saying, this stuff has energy. We need this stuff. Let's use this stuff. That in turn goes past the blood-brain barrier into the hypothalamus. And again, that's part of the energy pathway. So part of it's going to tell your body, yes, we like the taste of this. And part of it's going to tell your body, we need more energy. Now, having established all of that, how does that apply to your everyday life? And why is that the case? Well, in the past, as I said before, that could be an evolutionary model developed due to times of scarcity. If it's the winter and it's turning into spring, so really it's the phase in between winter and spring, it's the middle towards the end of March, and you stumble upon a berry bush that has just started growing or just became ripe. It's been winter, you haven't had any kind of carbohydrate source in a long time. Now you start eating away at those and it's a huge whack of sugar. You're probably going to eat a whole bunch more for two reasons. One, there's the energy component, the fact that you're getting a lot of sugar that you haven't had access to in a while, helping to feed your brain. That's going to tell your body to say, hey, that tastes pretty good. Find more of that stuff. Unfortunately, you're not going to be able to that often at other points. Had it still been winter, you wouldn't have had access to it. So what does that mean? Well, today, because of the overabundance of super palatable foods, things that taste good, well, it's very easy to overconsume them because your brain is still hardwired for a time when it wasn't abundant. Your brain's still hardwired for the time when it was a scarcity, when that berry bush was a rare occurrence. Today, that berry bush is everywhere. It's at the grocery store. It's at an actual, farm. It could be in your backyard. It could be anywhere. So, and that berry bush can be a billion other things. It can be Oreos and, and Reese's Pieces and stuff. So, moving forward, because again, we're not really focusing on sugar today. We're focusing more on fat. But basically, for instance, an oral stimulation with sweet solutions triggers a rapid, albeit modest, increase in insulinemia in absence of any ingestion or of a glycemic variation in rodents, and also likely in humans. The RNST also projects to a set of brain areas. Hold on one second. All right, sorry. Just wanted to make sure it wasn't anything important. The RNST also projects to a set of brain areas constituting the cortico-mesolimbic circuitry, known to be implicated in the emotional representation of food. So again, not only does food have a physiological component, obviously, as we know very well, it has an emotional component. That has to do with that liking, palatability, reward system. So, is integrated system, including the ventral tegmental area and nucleus accumbens, which constitute the reward system and are involved in the motivation to eat. The amygdala, the hippocampus, and hippocampus implicated in that learned memory, and the frontal regions of the cerebral cortex, i.e., prefrontal cortex, the orbital frontal cortex, and the cingulate cortex, which are responsible for decision making. By allowing instantaneous evaluation of hedonic value, i.e., pleasure or liking, incentive salience, motivation or wanting, and predicted reward value, i.e., memorization or learning of foods, this pathway plays a crucial role in eating behavior. Indeed, integration of orosensory signals, i.e., taste and somatosensation, by the reward system determines the palatability of foods that is to say their hedonic value. That is to say their hedonic value, i.e., the pleasure induced by some food in the mouth. Orosensory information also affects, in a lesser extent, the regulation of energy balance by the hypothalamus, i.e., the metabolic brain. During prandial period, while you're eating, the dialogue between the mesolimbic and the hypothalamus leads to decision makings, such as food selection. This two-step neural processing model highlights that the control of appetite depends on subtle balance between the right emotional brain and the metabolic brain. Dysregulation of this central homeostatic system can lead to obesity, whether hedonic hunger becomes the dominant compared to the metabolic hunger, that is to say, the real energy needs of the organism. So, breaking that down again, we kind of broke that down a second ago. If, let's say, there is an issue in terms of the circuitry of your emotional brain versus your metabolic brain, so your body only needs XYZ calories. Calories in, calories out, right? You know, okay, I only need 2,400 calories today. That's just a number thrown out. But because there is a dysregulation, because there is an issue with the emotional section, with the mesolimbic portion of your brain, you could very well go and eat 3,000. Now, one time you do that, if you do that one day, no big deal. You eat 5,000 one day. You know, Thanksgiving people do all the time, right? No big deal. But if you do that over time continually, then yeah, that's when calories in, calories out starts to matter. But it's not explicitly that you're just overeating for the sake of overeating. There could be an issue going on with the hormonal signaling in your brain. Now, post-ingestive signals triggered by the arrival of energy nutrients in the gastrointestinal tract provide additional information to the brain. It is noteworthy that the gustatory receptors first identified in the taste bud cells are also found in the small intestine. For instance, the enteroendocrine 1 cells express sweet receptors, T1R2, T1R3. I feel like I'm in Star Wars right now. They don't really express R2D2 though. Sorry, that was terrible. Let's stop with that. And release GLP-1 in the presence of glucose and in sweet receptor dependent manner. The satiety regulatory peptide can modulate eating behavior via the activation of GLP-1R signaling both in the enteric vagal nerve afferents, which are connected to the caudal MTS and the arcuate nucleus, a critical hypothalamic platform that integrates circulating signals of hunger and satiety, basically whether you're hungry or not. During the postprandial period, after eating, variations in blood macronutrient levels also contribute to the control of the homeostatic functions of the metabolic brain through the selective activation or inhibition of glucose and fatty acid sensitive hypothalamic neurons. So basically, after you eat, your body's metabolic brain, the one telling you whether or not we need more energy, changes. Either it turns on or turns off the hypothalamus saying, do we need more energy or no? Are we good? Do we have to keep eating? Are we hungry or are we satiated? So in conclusion, the progressive deciphering of the sense of taste reveals a complex system highly adaptable to changes both in the external environment, such as food availability, and body nutritional needs. The hedonic reward value of food appears to be closely linked to the gustatory system, raising the possibility that taste constitutes a significant driving force, leading to preferential consumption of highly palatable foods, often rich in sugar and fat. While mechanisms underlying the perception of sweet taste have been widely studied for decades, the putative role of gustation in the orosensory detection of dietary fat is only an emerging paradigm. Again, we know damn well sugar makes you eat more, does dietary fat, and that's what we're here for. I'll let you know a little secret. It does. Sugar, salt, and fat, put them all together, you're creating something that is clearly hyperpalatable. The food industry has known this forever, and if you look at anything that is a refined food, generally processed food, you're going to find all three of them in combination for that very reason. So palatability, or rather, let's talk about what we're talking about, a taste of fat, supportive evidence. So palatability is a major determinant of food choice and consumption in humans. Energy-dense foods rich in fats are especially palatable. Therefore, it is not surprising that preference for high-fat foods is widely shared trait in humans. In free eating situations, laboratory rodents also generally prefer high-fat foods to their standard chow. Orosensory detection of dietary lipids was long thought to involve textural and olfactory cues only. Recent data, however, strongly suggests that the sense of taste also plays a significant role in the spontaneous preference for fatty foods in rodents. Indeed, fat in mouth, it's a funny way of putting that, specifically activates lipid receptors to trigger a signaling cascade in the TBC. So that's taste bud cells. The resulting lipid signals use the gustatory nerve pathway to modulate both digestive function and eating behavior. In contrast, its role in human preferences remains to be fully established, mostly because the literature hasn't been done yet. About 70 years of refined food evidence that clearly shows that that's the case. But yeah, is it the triglycerides or the free fatty acids? This here they discuss whether or not it's the actual fats themselves or the full fats, long chain fats, or medium chain fats, or is it their breakdown components, triglycerides? From there, again, we'll skip through that for now. Next one is the influence of the salivary environment. Basically, since saliva plays a big role in the orosensory perception of dietary components, is it really important? Since 90% of saliva is produced by the parotid, with the remaining being secreted by the minor salivary glands, is saliva important? That's this portion here. Again, we'll skip through that for now. Chemoreception of long-chain fatty acids by taste bud cells. These are some of the potential mechanism pathways about how they're going to do it. So there are 4 plasma membrane proteins belong to 3 unrelated families, and displaying a binding affinity for free fatty acids have been directly or indirectly implicated in the chemoreception of dietary lipids by the gustatory papillae. So there's DRK channels. There are the GPR40. There's the GPR120 and the CD36, which I mentioned before. So, having established all of that, hold on one second. Having established that, again, this is one of those ones where if you're interested in really digging into the science on that, on the actual receptors and how they're actually transducting, you can reach out to me. I'll get this paper to you. But having said that, here we go. Let's move forward. So there's long chain fatty acid mediated signal transduction in taste bud cells. So the first one was more the free fatty acids, but there could also be receptor coupled downstream signaling, including phospholipase C activation and or calcium signaling and activation of the calcium channels. There could also be TPRM5 channels. So these here are ones that deal with the long chain fatty acid mediated. The first one had to do with the free fatty acids. There could be also they go into the neurotransmission of the fat signal, lipid signaling in taste bud cells. We have some nice pretty diagrams of those signaling pathways, which again is why we're skipping right through it for right now, just because we're already about half an hour in, and yeah, don't want to be on here all time, all night, on just one paper, because again, it is a pretty damn long one. So we have here some limitations to this particular model. We have whether or not it's shown efficacy in knockout mice, whether there's an actual affinity to long-chain fatty acids or a specificity for free fatty acids, whether there's lipid-mediated regulation, and then there's attraction for fat between those particular receptor sites. Here again is another variation on the model showing how that mechanism, how it can play a role. Basically, how that fatty taste can have an end net result in changing our eating behavior. Moving past through there, we have the compatibility model of our peripheral taste coding models. So we'll look at this model real quick. We have our selection and consumption of palatable foods. So in our hypothalamus, there's a homeostatic food intake. As it increases, the PFC goes and increases cognitive control attention. If that is down, that in turn leads to an increase in insula of the sensory and motor neurons. We have increase in emotion in the amygdala and hippocampus, and then we have an increase in motivation and reward in the NAC, the VTA, OFC, and the insula. So in a normal weight person, there is a down regulation there. In an obese person, it goes to the upwards regulation. So lipid and obesity, also a taste. Sorry, we'll look at concluding remarks regarding the actual pathways. In summary, some of the concerns about the candidates for gustatory lipid sensation raised here do not appear sufficient to exclude the fatty as a primary taste, at least in the mouse. In humans, there is a little evidence regarding the functionality of a free fatty acid transduction system in the taste bud cells. Additional studies are absolutely required to determine whether our eating behavior may also be driven by taste component devoted to the perception of dietary lipids, and what the health impact of such a detection system might be. So humans are very greatly in their oral fat detection. According to our present knowledge, it is tempting to hypothesize that this variability might influence our consumption of fatty foods, i.e. that hyposensitive individuals consuming more fat in contrast to hypersensitive subjects. Obese people tend to overeat fat-rich foods. The biological origin of the preferential food choice is complex. Emerging findings reveal that obesity may be associated not only with the reward deficiency syndrome, but also with dysfunctions in orosensory detection of dietary lipids. So, again, this one goes through a bunch more about how that could potentially be, but we're actually going to look at two separate papers specifically regarding that. So right now we're going to call it for this particular paper, and we're going to move on to this one here by Kindleysides et al., which is a newer paper from 2017. And what I'm actually going to do real quick is I'm going to edit the info, making a note that we're starting this particular paper around the 30-minute mark. All right. So down below in the description, you should be able to see now that we are doing this one. All right. So let's talk about this paper. We have perception of fat taste, aroma, texture are proposed to influence food preferences. Thus shaping dietary intake and eating behavior and consequently long-term health. In the study, we investigated the association between fatty acid taste, olfaction, mouthfeel of fat, dietary intake, eating behavior, and body mass index between 50 women who attended 3 sessions to assess their oleic acid taste and olfaction thresholds, the olfactory threshold for N-butanol, and subjective mouthfeel ratings of the custard samples. Of custard samples, not the. We hadn't established they were before. Murray, hey, what's up? Long time. Missed you there. So, moving forward, though, we have here. Again, if you have any questions, put them down. Put them on down. I want to hear them. But as we move forward here, all right, we are talking now. Whoop, let me pull that back up. Basically, what they did is they went and did dietary intake and behavior. Eating behavior were evaluated using a food frequency and 3-factor eating questionnaire, respectively. Now, again, you know how I feel about food questionnaires and all that fun stuff, but yeah. In this particular case, probably is the best way of doing that aspect of it. But binomial regression analysis was used to model fat taste and olfaction data. Taste and olfactory detection for oleic acid were positively correlated, as well as oleic acid taste hypersensitive women had significantly increased N-butanol olfactory sensitivity. The eating behavior, disinhibition, and BMI were higher in women who were hyposensitive to oleic taste. Dietary intake of nuts, nut spreads, and seeds were significantly correlated with high olfactory sensitivity to oleic acid, and these findings demonstrate a clear link between fatty acid taste sensitivity and olfaction and suggest that fat taste perception is associated with specific characteristics of eating behavior and body composition. So again, let's break that down. We have basically when people are hypersensitive, they had a slightly increased olfactory sensitivity. So they basically were able to sense those at a higher level or at a lower level. They noticed the fat. Their bodies were able to pick up on the fat at a lower level, so they were hypersensitive. Eating behavior disinhibition, which is a really backwards way of saying, oh, eating behavior inhibition, right? No, disinhibition basically meaning their inability to stop themselves. So their BMI and their inability to stop themselves from eating were higher in women who were hyposensitive to oleic taste. Basically, those women who were unable to determine that there was fat or required more fat to be present in order to notice there was fat in that food were more likely to be both overweight or at the very least have a higher BMI and were less likely to be able to self-regulate their eating habits. From there, dietary intake of nuts, nut spreads, and seeds were significantly correlated with high olfactory sensitivity to oleic acid. Basically, if you could smell it, okay, you were likely to eat more. These findings demonstrate a clear link between fatty acid taste sensitivity and olfaction. So basically, smell and taste can play a role. So we have here talking about the G protein coupled receptors. We mentioned that before, how sweet, umami, and bitter have been identified as those. Salt is recognized as a ion channel receptor, which is ENAC, and the sour taste receptor mechanism yet to be identified responds to the presence of acid. Now, we talked about in the first paper about whether or not fat is a 6 taste modality, that the G protein coupled receptors could be part of it. The ion channel receptors could be part of it. The CD36 in particular could be part of it. So fat ranges, though, creates a range of textural qualities which are considered to be well-known sensory properties of fats, such as creaminess, oiliness, and thickness. Till recently, it was perceived to be predominantly only the mouthfeel and olfaction, so the smell and the sensory qualities, but there's now considerable evidence that fatty acids can be perceived by specific taste receptors on the tongue, and several studies have demonstrated the gustatory sensitivities to fatty acids at low concentrations. So, as we established in the first paper we looked at, yeah, people actually might be able to taste fat. It might actually be a sixth taste. From here, though, we're going to look at whether or not, basically, the amount of those particular receptors you have, how much there is, can change the amount of food you can eat. So because being investigated now in the signaling pathways which govern the body's responses to incoming food. Nice, as you bake your holiday cookies. Nice. Just make sure that you don't go too crazy on the sugar, fat, and protein. Oh, sorry, on sugar, fat, and salt. I don't know why I said protein. And all the protein. And I know you're going to keep them nice and fatty. So that being said, it has been recently suggested that overconsumption of dietary fat might alter the sensitivity or the expression of taste receptors, and that high-fat diet exposure can decrease sensitivity to fatty acid taste in lean participants. These data suggest that sensitivity to fatty acid taste may have a significant impact on eating behavior and long-term dietary intake with important health consequences. So, commonly cited causes of obesity include major changes in our food environment, which have led to the overconsumption of inexpensive, hyperpalatable, highly energy-dense, and nutrient-poor foods. Given that fat-rich foods have a powerful hedonic appeal, preferences for fatty foods are important contributors to increases in body weight and metabolic disease. Now, I will add to the caveat to this that it is generally going to be the case that you're also probably going to be overconsuming carbohydrates too. But that's, a whole other conversation. The combination of high fat, high sugar as well, or high carbohydrate in general, particularly high sugar, is what's really going to be the long-term damage. Yeah, no, these are gluten-free vegan, but loaded with sugar, lol. Not for me. They're gluten-free vegan, not for me at all either. Definitely not. As soon as you said vegan, I'm out. As soon as I heard there's no eggs in there, gone. But moving forward, in the present study, we designed a series of experiments to investigate the relationships between fatty acid taste, olfaction, and mouthfeel modalities, and how fat taste perception measurements may relate to dietary intake and eating behavior. The present study aimed to measure oleic acid taste and olfactory detection, explore links between oleic acid taste, olfaction, and mouthfeel perception of fat, and investigate oleic acid taste detection and associations with eating behavior, dietary intake, and body composition. So there was a test that was used, and this study, again, just like the others, is down below. The first one is the only one that's behind a paywall, so you will be able to read this one on your own later if you want to look through the actual methodology yourself, but we're going to skip right through that for now. They also used some sniffing bottles of the fats to basically see at what level they could actually smell the fats, but yeah. So we have here our body composition profile. They basically measured anthropometric measures, including height, weight, and bioelectrical impedance analysis, so lean and fat body mass. They measured taste perception oleic acid, basically how much they tasted, how sensitive the women were to oleic acid, the threshold of whether they were low or high, which categorized them then as hypo and or hyper sensitive, well, would be hypo or hypersensitive to oleic acid or fatty acids. More on that later. The olfactory perception of oleic acid, basically, were they able to smell? At what point they were able to smell, as well as the N-butanol olfactory perception. Again, what point they're able to smell using the sniffing sticks and butanol. The mouthfeel perception, their subjective rating of mouthfeel. So, is it creaminess? Do you like it? How intense was it? Their dietary intakes, their daily energy, macronutrient, and food group intake, as well as their eating behavior. Were they restrained? Were they ravenous and didn't care? They were disinhibited. Or were they still hungry? And that was done using the SurveyMonkey online platform. So I guess the whole fat is more satiating idea doesn't apply here. So yeah, that's a great question, Murray. It kind of applies back to what I was saying a second ago regarding the sugar. If you're eating mono macronutrients, whether it be fat, protein, or sugar, there will be a point where you cannot keep eating. There will, unless you like completely severed your hypothalamus, which is, again, we talked about in the first paper, the metabolic brain. At some point, you're going to know there's too much energy here. Stop eating. And when you do it in a mono fashion, when you do it with just one macronutrient, that point comes a lot quicker because it's the same thing over and over. If it's and in Wired to Eat, which I know you've gone through, he goes through that really well. Rob Wolf really breaks that down. But the short version of that is that after a certain point, there's basically a loss of palatability. It's no longer enjoyable. So that brings back to the mesolimbic portion of the brain. If you no longer enjoy it, you're going to stop doing it. It's when you combine that fat, sugar, and salt that it throws our brain for a loop. It's like, whoa, we're not supposed to have all of these in one food. I don't know what's going on here. And it essentially becomes crack. Your body doesn't know how to process it. It has no idea what it's looking at. What we're talking about specifically here is people who are individually more susceptible to over-consuming fat simply because their body doesn't realize there actually is fat there. And this and the next paper really break that down. But essentially, hyposensitive people don't realize there's fat in a food. So they don't have a particular preference for a higher fat versus a lower fat. So you could give them, keeping the sugar the same rate, you could give them the same food as a high fat variety and a low fat variety, and they're not going to have a preference for it. People that are hypersensitive, those that can sense the fat at a lower level, are going to prefer the high fat one, and they're going to, as I said, they're going to consume it, and they're probably going to regulate themselves. Whereas those who don't sense fat as much are going to not inhibit themselves. They're going to not disinhibit themselves. Yeah, it's such a stupid way of putting it, because that's the way they keep wording it. They're going to not inhibit, so they're going to let themselves run freely and eat whatever the hell they want because their body does not process that, hey, or at least their brain doesn't process that, hey, there's fat here, there's extra energy, do something about it. So it's easier for them to overeat. So more on that in a second. But going through, again, here's the methodology, statistical analyses. From there, they break down the participants. We have a nice little waveform of the taste detection curves compared to the olfactory detection curves, as well as they are shown their taste model. So fatty acid taste hypo and hypersensitivity. Previous studies have defined oleic acid taste. Don't have oleic acid taste. It's probably a bad term or a bad time too. Oleic acid taste hypersensitivity empirically as a detection threshold, the lowest concentration at which stimulus is detected, determined by 3 consecutive correct taste trials, are given concentration. At a given concentration. At a concentration of less than 3.8 micromoles, that's the threshold. Reading past myself. In this current study, we used a model to determine taste detection rate. Interpolation of each detection curve was required to characterize participants as hypo or hypersensitive. So basically, if you were below the threshold of 3.36, to be able to determine below that, you were hypersensitive. And if you couldn't determine it until after 12.12 micromoles, then you were hyposensitive. Detection threshold, otherwise, so that was detection rate. Detection threshold was 2.58, excuse me. And then 11.10 was the hyposensitive, above that. Here's the probability in terms of taste detection. We have here basically their ability to detect different things at 0% coconut oil, 5% coconut oil, 10% coconut oil, 15% coconut oil of hyposensitive people versus hypersensitive. And basically, they had a comparison of their mouthfeel rating, their mouthfeel liking of high, of 15% coconut oil, medium, low, and no fat custard, split by taste amongst hyper and hyposensitive. So basically, Did they like it? Was it good? You see here that on the left side was it too dry? Was it just right? And what or was it too fatty or oily? Then we have here they disliked it extremely versus they liked it extremely. So in terms of those who were hyposensitive, those who didn't notice liked the ones that had higher fat content. The more of them liked the higher fat one. They weren't really able to detect it. So like, oh yeah, that's a good one. Whereas those that were hypersensitive liked it at a lower rate. So after a certain point, they're like, less of them liked it. Like, yeah, we're good. Less of them were apt to say, this is the one I want. When it comes to whether it's too oily, again, the hypersensitive ones, the 10%, 15% were like, whoa, too much, don't need this. Whereas the lower ones at 0%, they're like, oh yeah, it's about the same because they didn't notice it. So the more oil involved, the more oily the hypersensitive people were able to find. They were able to detect it at a better rate. So the discussion, getting here, the oil, acid, taste. Hyposensitivity to fatty acid and taste was associated with disinhibited eating behavior. You're gonna eat more if your body can't detect the fat. These findings of the study show remarkable parallels in fatty acid taste and olfaction detection rates with clear and consistent individual differences in detection ability. These individuals' differences in fat detection appear to be linked with disinhibited eating behavior that may have implications for long-term metabolic health outcomes. Okay, so the perception of mouthfeel. Again, you can read through this yourself, link in the description. So although the ability to perceive fatty acid taste varied markedly between participants, the association between fat taste and perception and disinhibited eating behavior observed in the present study suggests that fat taste perception may influence dietary habits that have long-term metabolic health consequences. Additionally, sensitivity to fatty acid taste was related to body composition, and hyposensitivity to oleic acid was clearly associated with higher BMI. In conclusion, our study presents strong evidence for a link between oleic acid taste perception, olfactory perception, and mouthfeel perception of fat, suggesting there are intimate relations between the multiple modalities of fat sensation in humans. Further research is required to investigate whether there is a causal relationship between fat taste perception and olfaction in the etiology of obesity, especially in an obesogenic environment of highly palatable, energy-dense, and nutrient-poor foods. So, we were talking before about the fact that in the past it was assumed or presumed that fat was the result of mouthfeel and the smell. This study right here in the 50 women showed that there was a high correlation between those and the women that were hyposensitive versus hypersensitive. So their ability from the chemoreceptors that we talked about in the first paper, the ability of those to detect fat is correlated with the mouthfeel and the smell. So people that are more likely to detect fat are less likely to detect fat are more likely to overeat it. And then finally, we have here some young, healthy Japanese adults were measured with their association of fat sensitivity with body mass, taste, and taste preference, and eating habits. This is one from last year, which again just helps to really send home the message that yes, so there's Asano et al. 2016 in the Tohoku Journal of Experimental Medicine. I'm gonna get kicked off YouTube for a stereotypical Japanese accent right there. All right, very insensitive. I know. Quick coastal craft kombucha. Sip break. Sponsored by myself and Sherry's Market. Should be getting kicked back for that. I drink the damn thing on every freaking live stream. I should be getting kicked back. Anyway, oral fat sensitivity may be related to overeating-induced obesity. However, it is largely unknown whether oral fat sensitivity affects taste preference and eating habits. Therefore, we aim to evaluate association between BMI, OFS, and the relationship of OFS with the 4 types of taste preferences: sweet, salty, sour, and bitter, and eating habits using serial concentrations of oleic acid homogenized in not fat milk and a self-reported questionnaire. So a similar methodology to the other paper, only now in Japanese men versus women. Versus, I believe it was just American women. Hold on. Let's see. Nope, wasn't American. Where the hell was the study done? Who were you women? New Zealand women. New Zealand European women. So basically white women in New Zealand. Alright. So now we have Japanese men. And they, whereas with the New Zealand women, they use custard and coconut oil mixed in. Here with Japanese men, they used non-fat milk, and they went and mixed in some oleic acid. And they also measured their sweet, sour, salty, and bitter tastes. Now, participants were divided into two subgroups based on oral sensitivity, using similar ranges. I believe it was 2.58 millimoles was the detectable amount for the hypersensitive in the other group, in the other study, whereas in this one it's 2.8. Close enough. The degree of sweet taste preference of the hypersensitive group was significantly higher than the hyposensitive group. So here we go. The degree of sweet taste preference of the hypersensitive group was significantly higher than that of the hyposensitive group, meaning those that were hypersensitive to fat, those that sensed fat at a higher level, were also likely to prefer sweets, meaning that if you notice fat at a lower level, if your body can tell, oh, there's a lot of fat here, you're also likely to prefer sweets. But furthermore, there was a significantly higher degree of preference for high-fat sweet foods than low-fat sweet foods in the hypersensitive group. Meaning again, if your body knows there's fat present, you're going to pick the high-fat ones rather than the low-fat ones. So again, if you know your body's able to detect fat at a lower level, you're going to say, I want the full energy one. But there was also a significant inverse correlation between the OA, or the oleic acid detection threshold, and the degree of both spare eating and postprandial satiety, meaning that your ability to detect, or the threshold at which your body is able to detect the oleic acid is inversely related. So it's negatively related with the degree of both spare eating, meaning you're going to spare eat, or just snack more, or eat more. Spare eating is a weird way of wording that. It's not a common way of wording that. You're going to eat more. You're going to overeat, and then postprandial satiety, you're not going to be as full after eating. Thus, oral fat sensitivity is not associated not only with BMI, but also with the preference for high-fat sweet foods and eating habits. The present study provides novel insights that measuring of oral fat sensitivity may be useful in assessing the risk of obesity associated with overeating in countries, including Japan, where BMI is increasing in the population because they're eating more of our crap. So, as we know, associated with a bunch of illnesses. We don't need to go through the illnesses. But indeed, obese subjects prefer lipid when compared with lean subjects, suggesting that inappropriate fat perception may influence the risk of obesity by affecting behavior. Therefore, it is valuable to clarify the possible role of oral fat detection systems in terms of weight maintenance in individuals. In this context, signals elicited by dietary fat, especially free fatty acid, are suggested to be initiated in the oral cavity and transmitted to the brain through nerves and neuropeptides and neurotransmitters such as beta endorphin and dopamine, which are released just after fat intake as the mechanism of the brain's reward system. And we talked about that in the first couple papers. It is well accepted that human individuals can perceive 5 prototypical taste qualities: sweet, salty, bitter, sour, and umami. And the process of sensing taste is first achieved at the level of taste receptor cells clustered in the taste buds. Again, we've talked about this a couple times. There's type 1 cells with the salty taste, type 2 with the sweet, umami, and bitter, type 3, type 4 with, we saw that pretty diagram in the beginning. In fact, just to send that one home, here we go. Here's our nice, pretty diagram of the different types of receptor cells. So we have our type 1, type 2, type 3, and type 4. Did they take into consideration what the subject's current diets were like? I.e., someone that's keto, let's say, body will have a different response to sugar let's say for someone who consumes sugar? That is a great question. I will come back to that because I have my own personal thoughts on that. I do not believe they did. That would be a complete change. But two factors to consider. One, this one here in the Japanese, I'd almost be willing to guarantee they're not keto. But, and this is a big one, in terms of why I believe those who are currently sedentary and overweight, who, as the study is saying, are likely to prefer fatty foods, will do better on keto. I think you can put together why that's the case, but I'll come back to that in the end. But yeah, I think that's a huge thing here. The fact that they respond less to fat, their body could very much play a role in the success of a keto diet. Now, having said that, will that change the body's ability to detect fat? Will a keto diet sensitize somebody to be more hyper or hyposensitive? That's a great question, and presumably it should, if it's not only a genetic component. So the part that I brushed through just for time purposes, gene protein coupling, the DRK, the CD36, it appears to be mostly genetic. But there definitely could be hormonal, and even more importantly, which is something else I consider, but again is beyond the scope of this particular video. We could do it again another week. But there could be a gut microbial component to it too. Your gut bacteria are absolutely telling your body to take in more fat or sugar. So there's a whole nother game to be played here. But moving past that for now, back to the point of this one here. I will address that more in a second. Sweet taste signals the presence of carbohydrate that acts as a source of energy. Salty governs the consumption of salts required for maintaining water balance in the body. Bitter protects against the ingestion of poisons. Sour taste signals the presence of dietary acids that are present in decayed foods and unripe fruits, as well as fermented foods like kombucha. And then umami taste helps identify protein-rich foods, which again makes sense since we consider that we're maintaining the other main macronutrients there: carbohydrates in sweet, water in salt, the proteins in umami, as well as in sour, we're talking about dietary acids, and in bitter, poisons. So those two are saying avoid bitter and sour for the most part. Eat things that have umami, salt, and sweet. It would make all the sense in the world that the other the other macronutrient, fat, should have a taste receptor built into our system. And again, it appears it does. So again, as we said like 4 other times, this paper is obviously, it's an intro, it's explaining the same thing. Up until recently, we didn't believe that was the case. Now we are starting to see here that there are possible lipid receptors, such as free fatty acid receptors and G-coupled protein receptors. Now, moving forward, previous studies which had examined the relationship between BMI and oral fat sensitivity, or the ability of individuals to detect fat in the oral cavity, were principally performed among non-Japanese individuals. Whatever. So now this one's done in Japanese. So that's to help investigate the literature gap here. Materials and methods, same concept. They used non-fat milk and added some oleic acid. So concentrations of oleic acid compared to the percentage of participants who detected the oleic acid. So as the concentrations got higher, pretty much everybody was able to figure it out. Here we have the detection threshold of oleic acid versus their body mass. The heavier people were, the higher the threshold was. The lighter they were, the lower the body mass, the lower the threshold was. So those who were hyposensitive, or rather those who were hypersensitive, those who knew when there was fat present, were like, hey, there's fat here. It's there. We're less likely to overeat. Whereas those who were heavier were more likely to be unable to detect that. Now, again, similar to your question, could their current diet change that? Yeah. Or partially genetics too. Maybe this guy right here, or girl, yeah, it's guy because they're all guys. Maybe this guy right here, even though he had a higher BMI, well, maybe he was also he also has the ability to detect that. Or Again, it's only a BMI 27. Could be somebody who's pretty muscular. So somebody could be somebody who's just highly muscular and is insulin sensitive and it has a regular functioning metabolism, and his body actually was able to detect the fat. But we'll come back to that. Over here, a combination of different tastings, so acetic acid to measure acidity or sourness, MSG to measure umami, sodium, so NaCl, to measure saltiness, sucrose to measure sweetness, and quinine to measure bitter. They diluted it down, and different ones there. From there, we look at our sweet food, our salty food, our sour food, and our bitter food. And what they did is they measured the food preference among hyper and hyposensitive participants. So they had the hypersensitive and the hyposensitive basically measure, what do you think about this? They then went to the food questionnaire. Some examples of the questions were, what do you think about the amount of your food intake? Is it extreme? Are you overeating? Is it adequate? Is it not enough? If you do not eat until your stomach is full, do you fail to feel a sense of satiety? I experience this frequently. So do you eat overeat? Do you like greasy foods? Yes, I like them very much. I like them moderately. I dislike them. How do you feel? How often do you consume sweet foods? I don't really do it. I do, etc., etc. So threshold for sugar, threshold for MSG, threshold for acids, thresholds for salty, and thresholds for bitter. Again, if you want to look through this, you can do that yourself. For now, it's kind of past the point of what we're looking at right now. Here we have a nice graph that compares food preferences degree to how much they prefer sweets, fats, low-fat sweet foods, high-fat sweet foods and low-fat sweet foods, salty foods, sour foods, and bitter foods between those who are hypersensitive and hyposensitive. And then we also have our comparison of hypersensitive to high-fat, low-fat, low-sweet foods, and then the bitter foods. And as we saw, in hypersensitive, they were more likely to prefer, statistically significantly likely more to prefer the high-fat, high-sweet food versus the low-fat sweet food, whereas those who are hyposensitive were less likely to really tell. And just as comparison, they put that right next to bitter versus sweet foods, and the hypersensitive, again, were more statistically significant. They were more likely to prefer the sweet food than the bitter food versus the hyposensitive. So that could be basically saying that in conjunction, the two of them combined are likely to play a role. The fact that they are generally hyposensitive to the hyposensitive to the fatty acid food means they're also more likely to have an issue with those sweet and bitters. But anyway, moving forward, we have here their questions. How do their degree of frequency of sweet consumption, their preference for greasy foods, how does that compare to the oleic acid threshold? But basically, in the present study, they clearly report that the detection threshold for oleic acid, but not the others detected in the oral cavity taste types, was associated with BMI among participants that consisted of young, healthy Japanese men and women. Oh, there were women too. Whoops. Moreover, more than 50% of the participants were able to detect the 2.8 micromoles of oleic acid in non-fat milk. Therefore, they divided the participants into two groups, oral fat hypersensitive group and oral fat hyposensitive group, to further investigate the data acquired. Regarding the examination and preference degree towards the other ones, they were divided. That's not really as important for right now, but in this context, it was noteworthy that the oral fat detection threshold was inversely related with the degree of self-awareness of both spare eating and satiety over food and after food intake. Because the results support the idea that oral oleic acid hypersensitive individuals have a greater ability to detect fat in sweets and other food products than oral hyposensitive individuals. Therefore, they can sense satiety after food intake more easily with the brain's reward system for fat signals and stop food intake earlier, eventually leading to the inhibition of excessive food consumption. However, a multidisciplinary model approach may be required for confirmation of these findings and clarification of underlying mechanisms in humans. The above results regarding the self-recognition of eating habits acquired from the present study may contribute to resolving the mechanism of modulation of OFS by recent fat intake as reported in studies conducted in Australia. So there's also the ones that were done in the last year in New Zealand. So let me read the last paragraph conclusion, and now we'll come back to that because that's going to address what I wanted to say about your question. In summary, we characterize the mode of association of OFS with BMI and the taste preference degree and lifestyle behavior and its effects on food intake in healthy Japanese young adults. Specifically, the oleic acid, so the fat detection threshold, but not the other 5 prototypical taste ins, was associated with BMI. Moreover, we found that oral fat hypersensitive participants, but not hyposensitive participants, but not hypersensitive participants, failed to show significantly higher preference for high-fat sweet foods compared to low-fat sweet foods on average. Basically, that those that were hyposensitive, those that had problems detecting fats, really did not tell a difference. They couldn't see which was better, the high fat or the low fat. They didn't really care. There was no preference for either. In addition, oral fat sensitivity had a statistically significant correlation with self-recognition of eating habits in terms of food intake and satiety after food, meaning those that were more sensitive were more likely to be aware that, yeah, I know how much I'm going to eat, and yeah, I know how full I am when I'm done eating. Therefore, in the future, it would be important to consider oral fat hyposensitive individuals as possible patients with dysfunctional oral fat detection systems and latent excessive food eaters in countries with increasing obesity rates, especially in Japan and the United States. Concurrently, because the reason for such individual variability is unknown, it would be highly valuable to clarify the underlying mechanism and establish a novel medical treatment or healthcare system, including lifestyle modifications for improving OFS. This could lead to prevention or recovery from excessive increases in BMI that are mainly caused by dysfunctional oral fat detection system. So, here we go. Let me synthesize this for you in answer to your question and to the paper itself. So, people that are hyposensitive don't know how much fat is actually in their food. Because of that, they are more likely to overeat, and they are less likely overall to prefer the high fat food versus the low fat food when it's in conjunction with the sweets. The sweets in that case doesn't really have as much of a role to be played. Now, as we established before, mono foods are less likely to be overeaten. Mono macronutrients, eating fat in isolation, is less likely to result in overeating than eating the high fat with that sweet. So, if you were to take somebody who was hyposensitive, even though they don't know how much fat is in there, if you were to feed them nothing but fat, their system, it takes them more to realize there's fat there. Their system is still going to get desensitized to it at some point and say, that is enough fat. Stop giving this to me. Cut the crap. I don't want any more. So that might be the way to get them to actually self-regulate. It might be a way to basically rewire their dysfunctional hardwiring in their brain. Feed them the fat, put them on a keto diet, particularly those that are sedentary and already overweight and or obese. There you go. That right there is how keto can work incredibly in that particular population. If we're dealing with individuals who are hypersensitive, they actually can taste the fat and know it's there. Those ones are less likely to be overweight as it stands right now, given the current literature. Having established that, maybe keto's not the best overall because they already are fat. They are sensitive. They know how much they can consume. Because of that, they're not overeating fat. Maybe they are overeating sugar. Maybe they have some sugar dysregulation issues. That's a whole different conversation than today. Now, moving past that, as I said before, the reasons behind it are highly individually variable. So yeah, there could be components beyond genetics. There could be microbial components, microbiome based. There could definitely be hormonal based. So that's a definite possibility. Keto could in turn potentially make somebody more sensitive. We know that consuming a keto diet and regulating your blood sugar and consuming more protein can make you more insulin sensitive, make you more sensitive to glucose administration. So there should definitely be a way of modifying your fat sensitivity. So yeah, could be something there. Long and short of that is if you're overweight and obese and sedentary, you're probably more likely to be over consuming fat in conjunction with other things. So hyperpalatable foods, foods that are designed to throw off your ability to regulate your appetite, are more likely to be problematic if you're already overweight. So it's a more, but at this point, we don't know if there's a chicken or the egg question, which one is the precipitating factor. I lean to think that it's a circle, that, people that are like that were already eating more could have already been dysfunctional, and then that cycle keeps going. A good way of figuring that out would be to do some studies, see if a ketogenic approach or a similar approach could actually go and take that cycle off. I lean towards it should be able to do that. You should be able to resensitize yourself, but the literature really isn't there yet. Or if it is, I haven't come across it yet. Because again, this is all pretty damn new research. But that being said, we are already about an hour 10 into this live stream. I was going to go through this recent paper about the the effectiveness of the autoimmune protocol diet for inflammatory bowel disease. It's actually the first one of its type with respect to uh, actually combating autoimmune disease. I mean, there's been a lot of empirical research and a lot of you know people doing it themselves. Just self n equals one self experimentation, but this one actually does appear. Why am I printing? I have it attached to a printer. Whoops. To appear to have some serious benefits. Unfortunately, because we are so far into this one, I don't think I'll be able to do this one justice in the time I want to. So I'm going to save this for next week. So this is to be continued. Yeah, long and short version though is that this one is in fact good. What I'm gonna do is I'm gonna leave that paper in the description down below. I'm gonna say is I'm gonna put it down at an hour 10 mark. Hold on. And I'm gonna say to be continued next week. All right. Having established that, I'll put that down there though. That way you guys can read that paper in the meantime if you're so inclined. But having established that, any last questions before we get going tonight? Because otherwise, we're gonna call it a night. And yeah, thank you so much for joining me. You know the deal. Like and subscribe. Please share it with your friends. Appreciate all this love and support you guys have given me. Beyond that, Fire Cream, they are the first run, are jarred. The labels are basically done. They've been sent off to the printer at last I heard. Hopefully, it's last I heard. They will be coming out soon. We are launching. It is finally actually happening. And that's kind of mind blowing, honestly. But yeah. So there's no other questions. That's it. This is my last show in my 20s. So, because after this one, my birthday comes up this week. So, I'll see you next decade, I guess. I'll see you next week, guys.